Thank you. So, hi everybody. Uh, we are live right now. Welcome to this series of innovation in hematology, which is being started by Dr. Nikhil and being carried on. And I know every third day or a fourth day we are having a meeting, Zoom link webinars. I think sometimes it's taxing also, but the whole idea is the information is being churned out from the West at such a fast pace, it becomes almost impossible to keep a pace for every disease and the information which is getting out to make the life of a people beautiful and more healthier. In that, keeping in mind, when we were looking at multiple myeloma space with Dr. Nikhil is the specialist for us, for our group. And Dr. Astha is doing a wonderful job for giving us the flow cytometry, MRD based regimes. We realize that there's no mention of a mass spec in Indian context. We do acknowledge that Nikita Mehra from Chennai, IDR, did some work on mass spec, and we also acknowledge Uday, who gracefully said that he has been working on mass spec, but except that, there's nothing which is happening in the space of mass spec. And to be honest, when I completed my DM, I probably would have never read about mass spec. I would have skipped my chapters on a mass spec as a technology, which has got a huge utility. And we are thankful to Thermo Fisher and Mr. Brijesh Pandey. On a very casual note, Mr. Brijesh one day told that there is 16 types of mass spec, and what are you looking at? And that was the revelation that I didn't knew anything. I mean, I don't know about many things, and we also don't know about many things. But that was a revelation in itself, and that's where I started chasing Brijesh. And thank you, Brijesh, after much of following you, because you're a great man and your in-depth knowledge has always fascinated us. I think that is very, very important, and that's why this the origin of this webinar took place through 2 months back. And we have got Dr. Srinidhi, we have got Rohan, and many people who have joined in today to listen what is mass spec as a technology and how it can be utilized. Because this will be a series of webinars where we'll try to understand the technological usage, and then later down where we can get it done in Delhi and all, we will come later to that point. But the promise to all of you is that by this year or this next year, we will have mass spec in Delhi. We will have multiple myeloma space being flooded with a mass spec because it is not expensive, easily doable, and the role of it in a non-invasive way, it is coming up in a big way. So with these thoughts, thank you, Nikhil, for starting this wonderful series, and all my colleagues who keeps on joining and spending their 1 hour precious time on these series. And I hope the content is of your stature and caliber, and we are able to take away many messages which can actually make our life beautiful, of course, more healthier, more scientific, and more knowledgeable. With this, over to Dr. Srinidhi. So, Dr. Srinidhi, are you there? Dr. Nikhil, are you there?
Hi, sorry, I got disconnected for a minute. Uh,
Yeah, over to Dr. Srinidhi.
yes. Hello, everyone. So, without wasting much time, I think I'll just introduce Mr. Brijesh, who has been very kind enough to agree to, talk about mass spec. And I think most of us don't really know much about mass spec, not even the basics. And he is going to make it easy for everybody in the hematology space, talking about the basic technology, how it works, what are the clinical applications. And Mr. Brajesh is General Manager Technical from Thermo Fisher Scientific, who have 16 types of mass spec machines, and we are really interested in which one are we interested in actually. And when he gave us choices, we were like, okay, now you need to teach us. So over to you, Dr. Brajesh, and hoping to learn from you a lot of it.
So thank you
Shared.
so much, Dr. Srinidhi. And first of all, I would like to really thank Dr. Bhargava despite all of your busy schedule and other things, you could figure it out time, and his progressive thought is very, very commendable. And I have a huge respect for him. Thank you, Dr. Nikhil, Dr. Astha, and everyone, and the participants all have joined today. Let me share my screen and then I'll take it forward to the mass spectrometer. The job which was assigned to me by Dr. Bhargava was very simple, that he wanted mass spec to be, excuse me. Yeah. So is it visible, my slides, Dr. Srinidhi?
Yes,
Yes,
yes, Brijesh,
yes.
it's visible. You're
Okay.
loud and clear.
Okay.
So
Thanks,
floor
sir.
is now yours for next 35 minutes to enlighten us with mass spec.
So I've been given a job to say the mass spectrometer made easy for hematologists. So I'm going to discuss a little bit brief overview of mass spectrometer. What are the different component of mass spectrometer? A brief theory of LC, which probably all of us know. A brief of fundamental of mass spectrometer, and then my emphasis would be more on the recent advancement in the mass spectrometry. And for all of you, I'm not going to bore with any of the slides from a corporate or organization. It would be purely scientific one. And I'm not going to discuss multiple kind of mass spectrometer. I have zeroed down to 2 or 3 models, whichever suits to you, depending on the need and experimental workflow. So the very first thing is that what are the mass spectrometer? So people who have not used the mass spec, they might have been wondering what these kind of machines are there. So mass spec is nothing. It's an analytical tool that measures the mass by charge ratio of a molecule in a sample to identify and characterize them. Now, what do you mean by identification and characterization? So these are the some of the major, in layman language we can say, the mass spec can identify an unknown compound. So in your case, if you have got a tissue biopsy, if you have got any serum sample, you have got any urine, anything, any biological fluid you have, there can be hundreds and thousands of analyte or compound. Now, this mass spec has got a unique capability to identify those compounds, say whether it is A protein, B protein, or what different kind of protein, or metabolites, or lipid for that. I'm talking in relevance with biological fluid. Now, if you want to measure that, okay, there are X and Y proteins are present. For example, IgG. What is the concentration of that particular protein or metabolites? It also has got a unique capability to determine the structure or chemical properties of a molecule. This property is more utilized towards chemistry people or towards the pharmaceutical companies who want to characterize a particular compound. So I'm not going to discuss on those parts, but yes, it can detect a trace level component or can detect the very finer concentration, and that's where it comes in the picture. Like you have got a multiple conventional technique, you have got electrophoresis, you have like gel electrophoresis, you have got some other alternate technologies, but what makes mass spec unique is that its specificity, its selectivity, and it's the lower level of detection. When we talk about lower level of detection, and that's where probably different model comes in the picture, is that it can go up to femtomole level or femtogram level kind of. So lower limit of detection. So that's where it comes a very handy or very useful tool, both for researchers as well as for biologists one. Now coming to this, what is LC and MS? Now, everywhere we say mass spec is a generic term. If you go in a little bit of scientific one, then people say that, okay, LC, MS. So LC stands for liquid chromatography and mass spec is a mass spec one. Now, why do we use the LC? Because you need to separate a particular. So once you have taken a blood, it has everything. It has got many components. Now you are interested into a particular protein, peptide, or metabolites. For that instance, you wanted to remove all other things except your protein or your metabolites. Nothing is of your interest. So you wanted to set. Now, so that comes as a sample prep. Coming to that, now within the protein, you wanted to separate different classes of protein or different types of protein so that you can identify them accurately. That is where the LC component comes in the picture. Now, before that, I just wanted to give you this schematics around that. So please keep this in mind that this is the basic schematics of any mass spectrometer, irrespective of any model, any vendor, anything here. So first is that you have got a sample inlet. Here in case of LCMS, you use liquid chromatography as a sample introduction tool. Now, in mass spec, there are three components. One is source region, another is analyzer, and then there is a detector. You can have this. So the first is that any compound which you can ionize, you can 100% assured that or rest assured that you can detect into the mass spectrometer. So ionization is the first part. Now after that, when you're coming, it comes to the analyzer. Analyzer, it separates or sorts the ions basis their mass by charge ratio. Then there is a detector which detects the signal, and then there is a software which has got an algorithm to give you or deconvolute that signal into the spectra. And this spectra, we deduce the required information. Now coming to that, what now that was a basic schematics. Now coming to that, what all can be analyzed? So as I said, if you can ionize a component, you can detect into the mass spectrometer, be it serum, urine, CSF, plant material, tissue, fruits, vegetables, cereals, anything you can detect into the mass spectrometer. As such, there is no limitation provided we can ionize a particular compound or sample into it. So this is an LC system followed by the mass spectrometer. This shows your ionization source. So there are different kind of ionization. There are multiple kind of ionization, soft ionization, hard ionization, and other things. But in case of biology, we only refer to the soft ionization kind of the thing. So what are those soft ionization technique? Generally, I have covered in my later slides that there are two different kind of ionization. One is basically ESI, which is electrospray ionization. Another is APCI, which is atmospheric pressure chemical ionization. These are the two different techniques how we ionize a particular molecule. Now, this is the HPLC component where I'm not going in detail, but we all know that it has got solvent, degasser, there is a pump, there is a column which separates analyte, and then we detect it in the mass spectrometer. And how it separates? In HPLC, this is basically you have got a stationary phase, you have got a mobile phase. Component of mixtures, they are carried through a stationary phase and their rate of migration. So those who actually interact with a stationary phase, the molecules, they will be eluted later on. Those who doesn't interact, they will be eluted faster. So once you have your mobile phase, you pass through the stationary phase, you get at a different time point. So here is a schematics which clearly highlights that you have got two components around that, A and B, and then you have got a column. You pass these compounds into the column, then they differ their rate of migration. So a particular compound elutes at a certain time, another compounds elute at another time frame. That's how we, so basis, this is called RT or retention time, basically how long the particular analyte has been there in the column or retained, and then elution time basis. Now coming to that separation, so this is another schematic. So you have got like all the different compounds, they are being eluted at a different time frame, you can see, and basis that you identify. So this is the first label of your confirmation that yes, every molecule has got a unique retention time. So you can actually get to know whether it is X or Y or Z compound which has been eluting at a particular time frame. That's there. Now coming to the mass spectrometer, as I said, it is an analyzer which measures the molecular masses. But technically, it actually measures the ratio of mass by charge ratio, nothing else. Now, there are different kind of mass spectrometers. So Dr. Bhargava and Dr. Srinidhi, they were mentioning different kind of mass spectrometer during my first conversation. So there are different types basis the analyzer. So one, if you recall my first schematics, wherein there is a source region, then there is an analyzer, then there is a detector. So basis the type of analyzer, mass spec can be of multiple types. A, mass spec can be of a single quadrupole or a tandem quadrupole. This is also known as a triple quadrupole. Then we have got ion traps. This entire category is known as nominal mass. So now there are two kind of nomenclature you can say. A, basis the mass accuracy. So if you see the upper one, left hand side, you can see these, they fall into the nominal mass. The lower side, left hand side, they all fall into the high resolution mass spectrometer or accurate mass. Now, as the word itself suggests that nominal mass and accurate mass, you can see that in the nominal mass, you will figure it out. It will measure the masses at a nominal level in terms of like, so if you have got a compound of 147.01, so up to 0.1 decimal, it will accurately tell you. But nominal mass. When you enter into the accurate mass analysis, it will give you up to 4 decimal. So you can actually go up to say like 147.01234 up to 4 decimal. You can accurately identify a particular compound. So that's the difference between a nominal mass and an accurate mass. Now, this is the type of analyzer you can detect into the single quadrupole, triple quadrupole, ion trap, multi, which is matrix assistance laser desorption ionization, but multi technique is now obsolete almost with the advancement of ESI platform. Then there is a QTOF, which is quadrupole time of flight equipment. Then there is an orbital trapping system or a magnetic kind of the system. In a nutshell, this is called nominal mass, this is accurate mass, or this is called quadrupole or triple quadrupole based system. This is called a high resolution mass spectrometer. I'm just giving a lot. Sometimes in nomenclature we do get confused, so that's why I'm giving you. Now coming to high resolution mass spectrometer, we have got different kind of portfolios. Explore is 120, 240, 480. There are different models. I'm not going into the details, but I'm just giving you a glimpse that yes, within a particular segment, you can have a multiple kind of models, and these models, they just differ on 3 parameters: sensitivity, resolution, and their speed. These are the 3 important parameter basis this. These mass spec differs. There is no other things. All basic fundamental remains same. Their functionality, workflow, everything remains same. Yeah. Now coming to the basics of mass spectrometer, how it works basically. So as I have said, ionize a molecule, then ions are sorted into the mass analyzer or detector. Mass analyzer, then signals are being sent to the detector, and we get a spectra around that. This I was talking about ESI, electro spray ionization source. So what do you mean by electro spray ionization source? So as if you recall my earlier statement, I have said that there are two kind of uh, ionization source: hard ionization and a soft ionization. ESI falls into the soft ionization. Why I'm talking about soft ionization? All biological data is being acquired into the soft ionization, because if you use the hard ionization, you will fragment into so many fragments that you cannot act, software algorithm cannot deduce the information. Now, using the soft ionization, you can actually get a more meaningful information with the help of software. I'll not go in detail, but just to give you a line that from LC, liquid chromatography, your flow is coming here. Then there are different gases, sheath gases, these are called. So your droplets are coming here. Now these are called these are heaters basically. So because in liquid chromatography system, your mobile phase may also come along with this. So all solvent need to be dissolved before these ions they enter into the mass spectrometer. At this point in time, it was not an ion, it was just a droplet. Now this droplet formation happens, then the evaporation. Because of these heaters, evaporation happens. Now, at certain point in time, there would be a ionic repulsion because their size reduces because of dissolution or a temperature, but the charges are still there. So then there is a phenomenon called Coulombic explosion. Because of that Coulombic explosion, the ions are formed, and these ions, they enter into the mass spectrometer. Yeah. So the ions, they enter into the mass spectrometer. This is called quadrupole region. So where you can select a particular ion, or you can scan a particular range basically. So these mass spectrometer, they are designed in such a way that either you can have a particular ion to be selected that only will enter, or you can define a particular range. Say like I'm looking for a mass range of 200 to 400, 300 to 500, whatever mass range you are looking for. Or you can tune, or you can ask through software that I am looking for a particular analyte which has got 257 M by Z, nothing else. So it will only select a particular analyte of that mass basically. Now, likewise, there is another ionization source which is called APCI, atmospheric pressure chemical ionization, where instead of the Coulombic explosion, here this is a corona discharge needle which actually ionizes nitrogen or oxygen in the source, and because of that, the ions are being formed. So let's look at that. Why we need different kind of ionization source? You must be wondering that why I'm talking about ESI, APCI, and other things. The reason is very simple. The different ionization sources being used basis their polarity and basis their molecular weight. So if you look at ESI platform, if you have got high polar or a mid polar compound, you can use electrospray ionization or a heated electrospray ionization as a preferred source. Now, if you see that there your molecule is of low polarity and a low molecular weight, then your preferred choice would be APCI. There is also a third ionization source, APPI, atmospheric this is photoionization, but generally we use ESI or APCI. This is a quadrupole theory. So I was talking about so quadrupole means there are four rods, and both the opposite rods, they have got a similar charges basically, and the ions are being selected here. I will not go in the details of that, but just wanted to give you how ions, they travel from, so the polarity actually changes, and because of RF and DC potentials, the ions move in the particular quadrupole region. This we should be knowing a little bit around that. So what are the different scan functions? So every in a mass spec, the workflow, you need to know product ion scan, precursor ion scan, or neutral loss scan. So you have selected a particular. So you have got, say like you have got a multiple ions from a source region entering into the quadrupole region. Now here you can select a particular ion, you can fragment that particular ion, and then either you have got the capability or choice either to take all the fragments, or you can select a particular fragment. So this is called a precursor or a parent ion. This is called product or a daughter ion. So this is how we should be knowing. So product ion scan means we are talking about the not the precursor ion, that ion has been actually fragmented with the help of collision cell, and there is a specific fragment ion is available there. And then we get a particular spectra, and for quantitation specifically, we actually need two unique transition for a particular analyte. Why we need two unique transition? Transition 1 will be used as a qualifier. That means this is the same peptide or this is same metabolite which we are interested in. And the second most intense peak which is there is used for your quantitation purpose. We have gotten standard, and basis that we can plot the linearity curve, and we can actually either quantify absolutely, that means if you have got an internal standard or a reference standard for a particular peptide, protein, or metabolite, you can do the absolute quantification. If you don't have the standard, then you can do the relative quantification. Both are possible. And generally, for a good quantitation, we need to have at least 8 to 10 data points in the UPLC scale. So till this, I was just giving you a brief overview for an uh, functioning of mass spectrometer, different components and other things. And let me enter into the main subject basically for which we are discussing. So for multiple myeloma disease, as we all know, and you guys know better than me, so it's an hematologic malignancy characterized by proliferation of plasma cells in the bone marrow, which releases extra intact immunoglobulin, and that is called as M protein. And these M protein has been well established as a biomarker for monitoring the multiple myeloma. Now, why there is a need? You all know that, whether it is like how the patient is being responding to the treatment or monitoring to the treatment, you can predict the particular disease relapse, or what is the response of a specific or a particular patient around that. All the current therapeutic treatment, they are basically invasive technique. And this technique, where we are talking about using mass spec, is the least invasive or almost non-invasive technique. So it doesn't require kind of like any unlike any other techniques. Well, so here we use a generic step. We just collect small droplet of blood, and then we identify a quantitypic peptide. So we actually do the sequencing. I'll share the next workflow. So this is like a personalized M protein monitoring. So what you need to do, you get a blood, and then you do the sequence. Now this sequence is very critical. Once you have got serum, you do the trypsin digestion, then actually you run into the mass spectrometer, you get a particular sequence. And out of that sequence, you get a quantitypic peptide, or that is also called as your clonotypic peptide, basically. That is patient specific peptide sequence. You get it. Now, you take the same serum or plasma, a separate one, and then you quantitate it. So you will see the difference of M protein over a period of time, whether it is going to be a progression-free disease, whether it is relapsing, or what is the status around that. So that's the basic workflow around that. Now, it is a little bit easier to be said than done, but critical thing is that having a particular peptide sequence for a particular patient. Now, here is the how your results look like. So this is your diagnosis sample. This is an XIC, extract ion chromatogram for a particular this heavy chain, which was a subject. So there is a study which is done by this. I've just shared this. This is a paper I'm referring. So they have done this study here, and then they diluted it for a diagnosis sample, and then they monitored the treatment. They have also compared with other conventional techniques. So you can see that serum protein electrophoresis, immunofixation electrophoresis, they have done it for, and then they also did it with mass spectrometer also. Now, when there was a patient sample, it was given the treatment with a particular drug, which we all know that it is used for multiple myeloma disease, and then they observed it over a period of time. So you can see they have observed at month 24 post treatment, and then also. So you can see that they have compared it, and it gives you a very nice correlation initially with the conventional technique. But if you look at later stages, you figured it out, the conventional technique are not so sensitive, or they are not quantifiable. And that's where the mass spec takes an edge over other techniques. So you can see that you can detect up to your 1.95 mg per liter. That's the lower limit of detection. And that's how you can see. Now you can see that the patient is safe or completely recovered. So this was an earlier one which was run on a very high-end platform, but over a period of time, with an advancement of science, the platform has reduced. So now this can fit into 2.5 feet by 2 feet of bench here. So this again the process remains same. So you take a blood, do the sequencing, or get a particular patient unique M protein sequence, and then you quantify them, and you can see that whether it is coming below the threshold or it is going up. Basis this M protein quantification, you can actually figure it out. Here it was an M protein dominant. Here you can see that after treatment, the M proteins are much less. So basically that decides you whether there is a chance of relapse or the patient is recovering one. So you can actually predict it. This like progression free survival or the chances of relapse. And this has been already certified. Some of the US labs, they are already doing it in their clinic. There are n number of publications and other things for with these applications. I can share these all for your reference. Then there is another company who is using it, this M site, but here the difference is that they are getting the M protein sequence from RNA-seq. So this is a bit of an inversion one. So they do a kind of uh, getting not from a serum but from your RNA-seq data. But this is also helpful because you already have an sequencer system in your facility. So I thought to just give you a perspective that if you can develop your uh, protein sequence or peptide sequence basically through that RNA-seq, and here also you can do the similar approach. You can quantify M protein. And over a period of time, so another 5 to 7 minutes, I'll take a very interesting tool. So mass spec pen in the hands of surgeon. And this fascinated me. So there is a group which is from Bayer College of Medicine. So Dr. Livia Eberlin groups. So they have developed in collaboration with Thermo Fisher and a unique mass spec pen, so which is intraoperative tissue analysis closer to the patient. So actually you can put the mass spec in the OT room itself, and surgeon can decide it immediately. They can be used as point of care to decrease the turnaround time of histopathology. Generally, we sometimes debate that whether these are cancerous tissues, non-cancerous tissues, and other things. And for that reason, Orbitrap Exploris was an obvious choice. A, because of its mass accuracy. B, its footprint. C, it's the accuracy basically, because the higher the resolution, better is the accuracy you will get it. So this is how it works basically. You have got a small trolley where your Orbitrap mass spectrometer is put there, and there is an uninterrupted power supply. Now UPS and everything is already there. Small pen is there, which is basically comes in the contact of tissue and takes the data. It just takes 3 seconds, just 3 seconds, and it will be able to give you a required kind of an information. So this, let me see if I can run this video. Sorry, the video is not there. So they are just taking, so you can see that this small mass spec pen, which is, this is an ovarian cancer, and they are able to detect cancerous, non-cancerous, and other things. Just see. On the back, you can see this is the mass spectrometer basically put here. You can see the thermo logo here. So mass spectrometer in the and now when I was going a little bit more deeper into that, I did realize that they have done some good publication also, wherein they have been able to differentiate different ovarian cancer subtypes, which is like high grade, low grade, or a normal ovary kind of the things, where they have used different kind of metabolites. Basically, here you can see the high grade one, which is PS and PI and ascorbate. However, in a low grade, they are talking about AI level as well as an glutamate level. So likewise, they have compared it data. The only one difference is that here the dependency would be that their algorithm. We need to submit the data to them, and there or we need to connect with the cloud, and then their algorithm, their software will predict us and give us a result within fractions of seconds. So that's a brief about mass spectrometer and its application in hematology. And I'm open for any question.
Wonderful, Brijesh. Wonderful. It's an eye opener for all of us. What kind of a technology and how the technology can make our life very easy for all of us. I think, uh, first of all, so you have to tell us as a novice, so what it goes. I mean, you have got reagents in NGS, you have got reagents in a CBC machine. So what goes in mass spec? I mean, just take a blood and put it into the mass spec, that's all, and the rest is done by the machine, or
No, no,
you
no,
need
sir.
a lot of human intervention.
No, we need a lot of human intervention. So this is not so easy. So first thing is that you take a blood, then you do the sample preparation and sample preparation, and then if the concentration is much lower, then we do the enrichment of the M protein. There are kits available. So once you do the say like you took 100 microliter of a serum, then you do the enrichment of the M protein, and which roughly will give you say like 80 or 100 microgram of enriched sample, and then out of that you take some like 20 or 10 microliter of that so sample, and you treat with a enriched sample, then you do the trypsin digestion. After trypsin digestion, you take out say like 20-30 microliter, and then you inject into the mass spectrometer. After injecting that into the mass spectrometer, rest of the work is mass spec. But the sample preparation is requires a human intervention.
So
So,
this sample preparation, the setup comes with your machine, or is it different, or is there an automation available for that?
There is an automation available, but these are generic ones. So every lab has got a normal practice. These are open one, and this is very very common. So
So it
trypsin
is something
digestion is
like
very.
library preparation before NGS.
No, library prepare here the library is not that, because in NGS that is a separate one. Here it is mainly the quality of a sample prepared. So as I said, apart from protein, we are not interested whether it is nucleic acid, whether it is something else. We are not interested into that. We are just interested into the peptides or M protein sequence. That's it,
so
and
what is
that's
the
where
sensitivity, Brijesh? What is the sensitivity difference between electrophoresis, serum-free light chain, and serum immunofixation? Is it better than, or the flow cytometry, it is better than all of these three, or it is equivalent somewhere in between? First question
it
is
is.
that.
It is
Second question
much.
is,
So this slide will give you an idea, sir. So this you can see that it's at least 3 to 4 times or 5 times you will be able to see the sensitivity enhancement in case of mass spectrometer.
wow.
So
And another thing is, does it give you a difference between the light chain and the heavy chain, or it
It
is
does.
only heavy chains, or the only heavy chains, or only the light chains?
No, it can give you both light chain as well as a heavy chain. But here for this matter, even if you are getting an heavy chain, that is good enough.
So, Dr. Srinidhi,
So Dr. Nikhil is asking, can we use it for disease monitoring if there is no baseline sample available? I was also going to come to the same question.
exactly, huh?
Uh, this is a difficult question to answer. Honestly, this would be an hit and trial one.
So I think what is my understanding for this question is that if you don't have a baseline sample, if you get a lot of oligoclonal peaks in the monitoring, then you won't be able to do that M by Z correction. If there is a correction which is done to rule out any false positive disease burden, if you don't have a baseline, you won't be able to do that correction. So I think it'll be very challenging if you don't have a baseline sample.
But you can use it from a healthy patient or something, and then you can try it. So,
No.
but it certainly requires a little bit of more
Yes.
experimental data to do that.
Yeah.
So my question is, many of the patients actually come to us for follow-up and they don't have their baseline sample or reports available. And many times, as we have seen, multiple monoclonal proteins would be there. For example, someone who's starting off as an IgA lambda on follow-up may have IgG lambda, IgG kappa, multiple monoclonal bands. So very often, if you don't have an analysis of the baseline, will this still be helpful to look for disease monitoring as a marker in peripheral blood MRD?
Yeah, so that's a very interesting question
I
you asked.
can you hear me?
Yeah.
This is Brigadier Tathagat Chatterjee. Am I audible?
Hi, sir. Good evening, sir.
Good
Hi.
evening, sir. Welcome, sir. Welcome.
Hi. So this is a beautiful presentation by Dr. Brijesh, and I'm just sharing my experience because I've worked on this with the National Center for Cell Sciences and Ganeshkand, Pune University. So we had to have these cell lines and the baseline preparation. So we analyzed a hell of a lot of normal samples and then compared to the multiple myeloma patients at diagnosis, at induction, at remission, and at relapse. So you need the baseline comparison, without which it matter becomes very, very difficult.
So,
That's it.
Dr. Brijesh, can I ask you that, for example, in NGS at least, if I don't have a baseline and if I'm asked to do an MRD, I go back to the baseline bone marrow aspirate slide, I scrape it and I use the DNA from there. Is there a possibility to get to run the protein on tissue?
Yes, it is possible. But as Dr. Chatterjee said, and probably Dr. Chatterjee is referring to Dr. Shrikant Rapole, who is using our system. Am I
Yes,
right, sir?
Srikant Rapole. Yes,
Yeah,
absolutely. He was my
yeah,
co-PI, Srikant
yeah.
Rapole. Correct.
So I know, and we know each other well. So my point is that it it's basically, for the moment you enter from a research to clinician sites, there is a lot of validation is required. Once we have got an enough number of patients, we can actually get to a conclusion, Dr. Srinidhi. So from technical point of view, yes, it is doable. But if you ask me, had it been validated, what are the false positive rates and other things, that's where the the challenge comes. We need to actually look into the uh, large number of data sets, and I believe that the hospitals like yours, where you have got an alternate conventional techniques or alternate like NGS and other platforms are available, this makes sense that you can have over a period of time, you will have a very good data sets to compare both of these things.
Yeah, because right now the concordance between flow and this is 67 to 68% only. So
Actually, it took us a lot of time, as Dr. Brijesh said, when we validated our first experiments, and without which we couldn't proceed. So we had to buy these cell lines from USA, and we had to validate all the normal proteins vis-a-vis the proteins you get in myeloma and the aberrations between the two. So it took some time.
is there some baseline? Is there some baseline over which like, do we have to run normal healthy controls first to form a baseline?
Absolutely.
Yes.
Absolutely. Absolutely.
Minimum kind of a thing, and then only above that would we
Yeah. So
have
that took us
normal?
1 year. As Dr. Pandey would say, it took us 1 year to validate normal samples, and then we started the next year on the myeloma patient. So it took us some time.
Does it work something analogous to copy number validation?
No, it is not. It is not that.
No.
No,
It's
it's not that. No, it's not that. Nice.
Yeah, because
But
the validation is very tedious, so I
yeah, it is.
Do you think that this is going to reduce the cost?
think
From an Indian
particular
perspective, Brijesh, we heard that this is going to reduce the cost in comparison to immunofixation, which costs 5,000 rupees now. in your great experiences, is the mass spec ready to bring down the cost of monitoring in patients of multiple myeloma? Because multiple myeloma is a chronic disorder which entails a huge load of burden, economic burden onto them for repeatedly doing the test again and again. So what do you feel about that?
If there are two elements, if the sample load is less, this might not be economical, but if your sample load is higher, this is going to be an economical for sure compared to the conventional one. Now, in the conventional one, it all depends like which technology we are comparing. Maybe the electrophoresis or immunofixation. If you're saying that, and is this 9,000 is the patient cost or actual cost, sir? You are
1,000 is the
referring?
patient cost.
So
So I'll tell you why, because the Iceland did the screening
Next
of
episode.
all
Our
entire
morning topic.
population, thinking as an ice stop myeloma. They screened all the population on mass spec to know whether it is MGUS, SMM, or multiple myeloma, just to identify the M band. So it means that the whole village, I heard that Uday has been doing the same thing in the country. Dr. Tathagat can enlighten us if he has the knowledge, but that's what
No,
we heard. So it
no,
means that
it
it
is.
has to be.
Yeah, you're right. It is institutional based now, and as Dr.
ആന്റി
Brijesh said,
ഒരു ഇഞ്ചക്ഷൻ എടുക്കണ്ട കേട്ടാ ചെറിയൊരു
it
വേദന
is
ഉണ്ടാവേ
someone is talking in between.
Yeah,
It is
Roy, can you
institutional.
unmute?
Hmm,
Can you mute
yes,
yourself, Roy?
yeah. So it is institutional based, and the more the samples, the more economical it will become. Right now, we have got this mass spec in NCCS where Uday is working with FMC and NCCS in MOU, which we had done in 2014. So we have done a few cases. We have, in fact, done bortezomib resistance also by mass spec. So a lot of things are coming up, but right now in patient care, I'm not very sure how economical it will be. You are the best one to tell me that.
Brijesh, you have to tell us.
as I said, as of now, this is not so economical compared to this. We can optimize it and bring it, but as you said, 9,000 is a patient cost. If you're referring, say, like anything less than 5,000, it is not going to be economical. It would be roughly around 7 to 8,000.
Very true, very
It will
true.
be, yeah. And Brijesh, which is the best thing, which is the best machine for a hematologist to use for multiple myeloma?
As I said, Exploris 120, small footprint, fast system, robust. You can use it for a. The only challenge would be that for a sequencing and other things that also works well. So and cost is also very effective. So I think Exploris 120 is the one of the best model in my views.
And does
Yeah,
it have
true.
a AI-enabled software in analysis or something?
Yeah. So software is common for all the mass spec, all variants. There is
No,
no difference.
no. So once we had collaborated with this third party, and they were giving an AI-enabled analysis of all the proteins which were detected just to predict, response and predict survival. So does this have that option, or how is it? Because that
No,
particular
so
machine.
let me give you clarity. So MassPen, last one which I have shared, uh, that is a separate one. There the costing is not there. However, rest of the other things, whether you are looking for like doing the sequence and then doing the quantitation, it is an one-time small investment. Then you have a software in your lab itself. You need not to go anywhere else, and you will be able to do it. The major cost is the upfront capex. Running cost is reasonable. Rest of the things is reasonable, and softwares are perpetual license software, so you need not to worry about.
Yes, Nikhil last time showed us to the group that their papers published in JCO, which has compared the next generation flow with mass spec from the peripheral blood, and the sensitivity for picking the MRD is almost the same. So, I mean, if the MRD in the myeloma space for the flow cytometry is yet to gain its credence like ALL, actually the mass spec can actually help in reducing the dependency uh, on the bone marrow itself. So you see, the bone marrow flow cytometry itself can cost between anywhere between 12 to 14,000, and it cannot be done every time because it's more invasive in nature. So if this technology can be established in Delhi with the help of Thermo Fisher, I think it will be a boom for everybody, because then things can actually be taken care at the peripheral blood. From the peripheral blood, it can be taken care of and it can be done repeatedly.
Yeah, this is a very good point because when we did our experiments on multiple myeloma proteomics, we compared peripheral blood and bone marrow, and we found equal, effectiveness of the peripheral blood. So we call them biomarkers. So we found out biomarkers in the peripheral blood, so you didn't have to do the marrow itself for basically for diagnosis as well as maybe in the future MRT.
Yeah.
Yes.
Tathagat sir, I have a question. Am I audible?
Yes, yes, yes.
Nikhil.
Tathagat sir, my question is, in your studies, whenever you had done proteomics data for myeloma, did you really use mass spec as an MRD marker? And if
No,
so,
no.
what was
No.
okay?
We didn't use MRD marker at all. We used it for validating the proteins in the normal versus the proteins in the myeloma patient. What was the
Myeloma
difference in the proteins between a myeloma and a normal patient at diagnosis, between a myeloma at diagnosis and a myeloma at relapse, and between a myeloma at diagnosis and a myeloma at remission? That was the experiment. MRT, we have, I think Uday will be doing MRT now.
So
We
from
were
the
scared.
data,
Yeah. We were
if we
just setting up the lab for myeloma proteins. How, in what way are they different from the normal protein? So we bought those cell lines from USA and we validated it.
Sir,
Understood,
one
sir.
more important thing is the amyloidosis part is missing here.
Yeah,
I
and
need
that
to
is,
understand
yes,
from Brijesh is Mr. Brijesh is that can we actually characterize amyloidosis, because it's a protein transthyretin and between amyloidosis, AL amyloidosis versus fibrinogen type, like I mean every year the classification for amyloidosis is ever increasing. So can we use that here also?
Yeah, so uh, yes, it is possible, but I need to authentically share this. I need to search for the literature and share with you. However, differentiation or characterization is very much possible, and that's what people do in a biopharma space. So if we have got different these peptide or monoclonal based drugs which are coming up, so that's how they do it basically.
very true. And this particular thing, Dr. Brijesh, is being taken up as an ICMR project by GB Pant, the mass specs in amyloidosis. So this is coming up in a big way.
And just to add on, in patients with IgG kappa multiple myeloma on daratumumab, very often with conventional techniques, it becomes very difficult to distinguish the monoclonal protein from the abnormal M protein. So this is a very good tool to differentiate this as well.
So I think what I'll do is I'll share I think they're all there in the net, Journal of Proteomics and all. There are 6 papers which came out since 2014 from NCCS and AFMC when I was there. And a lot of preliminary data might be useful to you. We have not worked at all on amyloidosis. That was not in our,
And so
horizon.
the
Those
whole
days,
idea is that
yeah,
we need to bring this machine quickly to Delhi and then can
yeah, yeah,
make
yeah.
this center for excellence of for Delhi amyloidosis group or Delhi
Very true.
mass spec group. So we just want to do that. And that's why the understanding we want to understand is what has been done in the country and what
Yes,
has been
yes, yes.
offered as a clinical service. So we might have done huge things in GB Pant and all, but
No,
as a clinical service,
GB Pant has not started. They're planning to take this.
yeah, as a clinical service, our job is to bring the clinical service, and that's what Dr.
Absolutely.
Nikhil has been after in myeloma space is a clinical services which can be in an affordable price can be offered to the patient.
Dr. Rahul, I'll share all my data papers as a preliminary with Srinidhi. Maybe this will create some understanding of this subject better.
Perfect, sir.
Can
Perfect.
it run on any body fluid or is it like
Any body
No,
fluid.
any body fluids. Right now, the NCCS is doing AML, interstitial fluid, with Uday in FMC, a mass spectroscopy. So they have now left myeloma and now they have gone to AML. So a lot of things are happening. Yeah, I'll
So,
share my data with you. Yeah,
sir, AML, if the count is very high, won't it become diagnostic?
AML. मेरा एक्सपर्टीज नहीं है माइलोमा में बोल सकता हूँ.
ठीक है.
I worked
Perfect.
on myeloma proteomics.
No, I think Uday and Army is doing a great and wonderful
Yeah,
job
yeah.
for the
Yes,
in the
yes,
service
yes.
of the nation. The way they have defended our borders. Similarly, the physicians from Army continues to defend the pride of us in by producing the high quality science. Thankful to Tathagath sir, and under his leadership and under his observership, we have completed 50 bone marrow transplants in ESI, which is a feat in itself. How we have started, it is him who brought us in, got everything established, and he kept a constant vigil on nothing going wrong. So the best part is Dr. Tathagat sir supported us, but kept a silent watch from the background, from a umbrella view that nothing goes wrong. And with that, we have been able to do successfully 50 transplants. Thank you, Dr. Tathagat, and we come to an end of this meeting. Great.
And
Tathagat sir, any parting comments?
no, just I wanted to compliment Dr. Brijesh. It was a wonderful presentation.
Thank you so much. Your words are it acts as a catalyst for me to do a little bit more. And I'm very happy and impressed with the kind of work Dr. Bhargava and his team is doing. Honestly
True.
speaking, before that, no one has asked to think or thought on those lines. And probably this is going to be the first center in India who would be doing a patient sample. So far, all the work is being done just for a research. So I wish this materializes fast.
True,
You
true.
have to make it materialize.
There are
Thank you,
many
Asta.
elements, madam.
Thank you, Asta. Thank you, Nikhil. Thank you, Tathagat sir. Keep
Welcome.
the blessings on our head and keep us guiding us. I think the best blend is of enthusiasm and the experience. What Tathagat sir brings is both. We don't have experience, but we have got enthusiasm, and Dr. Tathagat sir complements it very well. So thank
So
you for all your guidance, and thank you everybody for joining in yet another successful webinar on innovations in hematology. We'll see you Friday when Rahul Banerjee will be speaking on. Dr. Nikhil will be quizzing him on the toxicity, how to manage toxicity in the myeloma space. So looking forward on a Friday evening again with Rahul Banerjee from US, from Seattle.
Wow, lovely.
Thank you.
Thank you so much. Thank you. Bye. Good
Thank you,
night.
sir.
Thank
Good
you,
night.
everyone.
Thank you, everyone. Have a
Thank
great evening.
Thank you.